Okay, so welcome to this next video in the Pharmacology of Transporting Epithelial playlist. And this video, what we're going to study is the Cystic Fibrosis Transmembrane Conductance Regulator, which is quite a complicated anion channel. So the Cystic Fibrosis Transmembrane Conductance Regulator. So it's got a massive, great name, and it's often usually abbreviated to CFTR. I don't know what happened to the conductance, and so some people often just refer to CFTR as the Cystic Fibrosis Transmembrane Regulator, but its full name is the Cystic Fibrosis Transmembrane Conductance Regulator. And basically what it is it's an anion channel. A channel which allows anions, which are negatively charged ions, to pass through it. Now, one of the main ions which passes through it is chloride. Chloride has a negative charge, so it's an anion, but there are others that also pass through it. Now, in this video, what we're going to do is we're firstly going to study the structure of the Cystic Fibrosis Transmembrane Conductance Regulator, and then we're going to study how you actually open it, because it is not just a channel that is open all the time. There is a series of events that cause it to open, and they're quite complicated. It is not a simple channel by any stretch of the imagination. So, we'll begin with its structure. So basically, it consists of two transmembrane domains, which both have 6 spanning alpha helices, making them up. Okay, so you start off down here, basically, with the amino terminus, and it is one polypeptide that makes up the entire Cystic Fibrosis Transmembrane Regulator. Oh, I've missed out conductance now. The Cystic Fibrosis Transmembrane Conductance Regulator. So you start off with the amino terminus of the polypeptide down here, and then it has an alpha helix which spans the membrane, and then it has another alpha helix which spans the membrane, and it continues on, and it does it 6 times. So, like that. So it ends up with 6 alpha helices which span the membrane, and that is known as one transmembrane domain. So I'll call this transmembrane domain 1. Okay? All of that, all of those 6 alpha helices which span the membrane are known as transmembrane domain 1, or TMD1 for short. And the individual alpha helices, if you want to label the individual alpha helices which span the membrane, you label them T1, then T2, T3, T4, T5, and then finally obviously T6 here. So basically, the transmembrane domain 1, TMD1, consists of 6 membrane-spanning alpha helices labeled T1 through T6. Then what you have is a loop connecting this to a second transmembrane domain with 6 alpha helices spanning the membrane. But there are some important domains in this loop. There is what is known as the nucleotide binding domain 1. So I'll put this here: nucleotide binding domain 1, okay? Binding domain 1. And I should have drawn that with 2 sort of binding sockets there, so I would like another indentation there. Okay, nucleotide binding domain 1, and obviously that's also abbreviated often to NBD1. Okay, and then you have a second important domain in this loop connecting the transmembrane domain 1 to the transmembrane domain 2, which is the R domain, or the regulatory domain. So this is the regulatory domain, and it's often just abbreviated to the R domain. And then we enter the second transmembrane domain. So we have, again, 6 alpha helices which span the membrane here, and they make up the second transmembrane domain, so transmembrane domain 2. And if you're going to label these membrane-spanning alpha helices, you label them T7 for this first one, then T8 for the second one, T9 for the third one, T10 for the fourth one, T11 for the fifth one, and T12 for the final one, so it's T7 to T12. And then finally what you have is a domain called the PDZ domain here, and finally you end with the carboxyl terminal of this polypeptide. And that pretty much is the structure of the cystic fibrosis transmembrane conductance regulator. Oh, sorry, no it's not. I've missed out something crucial. You have another domain here. Before you have the PDZ domain, you have another nucleotide binding domain, which has again 2 of these sockets in. So this is the second nucleotide binding domain. I do apologize for that. This would have gone horribly wrong if I hadn't put in that nucleotide binding domain. So let me highlight the nucleotide binding domains, because they are extremely important in deciding when this channel is open and when this channel is closed. Okay, right, so there we go. There are the 2 nucleotide binding domains, the nucleotide binding domain 1 and the nucleotide binding domain 2. Right, so I want to explain firstly what this PDZ domain does, then we'll discuss what the regulatory domain does, and then finally we'll discuss the nucleotide binding domains and how they control when the cystic fibrosis transmembrane conductance regulator opens. Okay, so firstly this PDZ domain. PDZ is something that you will see often if you study biology. It's basically a combination of amino acids. It's a sequence of amino acids that shows up all over the place, and the reason it shows up all over the place is it is used to bind to cytoskeletal molecules. So the cytoskeleton consists of loads of microtubules, which basically form the solid core of the cell. Because if you think about what a cell is, so if we have a cell here, the cell membrane is this fluid, gooey mass. So that doesn't give the cell a very pronounced structure. So in fact, what actually holds the cell up and maintains its shape the same is that you have a meshwork of hard proteins which are holding the membrane up. Basically, they hold the membrane and keep the cell in a conserved shape, basically. So this PDZ domain is basically the way in which this channel attaches to that cytoskeleton. So this is the cytoskeleton, and there are loads of different proteins which contribute to the cytoskeleton, such as microtubules, which are made up of tubulin. Okay, so basically PDZ is a combination of amino acids. It's a sequence of amino acids that likes to bind to the cytoskeleton and therefore attaches this protein to the cytoskeleton. Okay, so that's what PDZ does. Now let's talk about what the regulatory domain does. So the regulatory domain is the site of phosphorylation by protein kinase C and protein kinase A. So if you remember back to when we were discussing secretion of salt, we discussed how there were stimulatory signals that provoked the epithelial cell into starting to secrete salt. And one of those was that you had agonists which were causing a rise in cyclic AMP. So let me just remind you of these pathways. What we had is so let's say if this is an epithelial cell over here, so let's go over a few of these pathways. The pathway by which we secrete salt. So one of the pathway by which we secrete salt was stimulated. One of the stimulatory pathways involved agonists at G proteins which were coupled to the alpha S G protein, so GS. So you had some sort of agonist to this G protein coupled receptor. And this G protein coupled receptor was coupled to the G protein GS, basically. So here is the alpha S subunit, and here is the beta gamma subunit. And alpha S is bound to GDP. And when the agonist binds to this GPCR here, the GPCR's catalytic activity turns on, and it cleaves the GDP off from the alpha S and binds GTP in its place. So you get an alpha S GTP molecule instead, okay, like that. And alpha S GTP dissociates from the beta gamma subunit and goes off on its own, and then activates an enzyme which is in the cell membrane, basically, which is the adenylyl cyclase enzyme, which looks kind of like this. So it has 2 transmembrane domains, similar to cystic fibrosis transmembrane conductance regulator, and each with 6 membrane spanning domains, in fact very similar to this. And this is the enzyme adenylyl cyclase. Okay, so the alpha S GTP molecule is going to go and activate the adenylyl cyclase molecule, and adenylyl cyclase enzyme, I do apologise, and adenylyl cyclase will convert ATP, adenosine triphosphate, into cyclic AMP and pyrophosphate as well. So you get pyrophosphate being given off as well, so pyrophosphate. Okay, and basically cyclic AMP is going to activate protein kinase A, and protein kinase A at the time when we studied secretion of salt, protein kinase A, we just drew it as activating the CFTR basically. So we drew it here, activating CFTR. So let's just draw CFTR here. We just drew it as a channel like that before, and just wrote CFTR like that. So protein kinase A was activating CFTR. Well, basically, this regulatory domain is where it actually phosphorylates. So, we also said at the time when we were studying secretion of salt that it was slightly more complicated than just having protein kinase A. You also had to have protein kinase C. And if you just have protein kinase A, under experimental conditions, you can just have protein kinase A and have no protein kinase C anywhere. You do not get activation of this CFTR. So basically, now let's have a look at how that works. So basically, this regulatory domain here is phosphorylated by protein kinase C. So protein kinase C comes along and phosphorylates this regulatory domain here. So adds a phosphate group onto some part of this regulatory domain. And that changes the conformation of the regulatory domain and makes available a phosphorylation site for protein kinase A. So basically, you need the phosphorylation by protein kinase C to happen first in order to have an available phosphorylation site for the protein kinase A. Once this available phosphorylation site has emerged, because you've phosphorylated it by protein kinase C, protein kinase A can now go ahead and phosphorylate this regulatory domain. Now, this new phosphorylation site on this regulatory domain. And if protein kinase A has not phosphorylated this regulatory domain, the channel will not open. So you have to have protein kinase A phosphorylation of this regulatory domain in order to get the channel to open. And if you want protein kinase A to phosphorylate the regulatory domain, you have to have had protein kinase C phosphorylate the regulatory domain. So both of these are required even for the channel to even consider opening, basically. So, but I should stress, protein kinase A phosphorylating it does not make it open. It's still closed at the moment. It's just that it cannot open unless protein kinase A has phosphorylated it. We will go through the steps that actually make it open, but those are totally futile unless the regulatory domain has been phosphorylated by protein kinase A. Okay, so what did I want to say? I've forgotten what I wanted to say, so we'll move on to the next bit, which is how this channel actually opens now. So the next bit, I know what I wanted to say. I want to tell you about the other name for the CFTR. So often what people will call CFTR is they will call it a cyclic AMP activated ATP gated anion channel. You will see it referred to as that, basically. And when this bit here, cyclic AMP activated, what they mean is cyclic AMP activates protein kinase A, and then protein kinase A activates this channel. So it gets it ready to open. If you don't have cyclic AMP, then you don't have protein kinase A, and if you don't have protein kinase A, this channel cannot even open. So that's the origin of this cyclic AMP activated portion. Okay, now we're going to go on to the ATP gated, because ATP is what actually makes this channel open, basically. So, the first thing that happens is that, and I'll draw another picture down here, basically. So if we just have a bit of a recap of the structure again, so that I can draw another picture out. So you have your amino terminus down here, so a bit of revision. The amino terminus down here, and you have your first membrane spanning domain. You have your second membrane, you have your first you have your first alpha helix which spans the membrane, second alpha helix which spans the membrane, third, fourth, fifth, sixth, and that completes the transmembrane domain 1. And then you have this loop which has the nucleotide binding domain 1 here. So here is the nucleotide binding domain 1. And then you have this regulatory domain over here. And then you go into the transmembrane domain 2. Which again has 6 membrane-spanning alpha helices. So this is transmembrane domain 2. And then finally you have the nucleotide binding domain 2 here, and ending with the PDZ domain, here, PDZ domain here, and then finally the carboxyl terminus down there. Right, so I'll just label the nucleotide binding domain 1 and the nucleotide binding domain 2. So let's assume that we now have phosphorylation of the regulatory domain. So we've got phosphorylation of the regulatory domain by protein kinase C, and we've also got phosphorylation of the regulatory domain by protein kinase A. So that bit's all done. Now we need ATP to come along and actually cause this channel to open. So ATP basically binds to the nucleotide binding domain 1. So I will draw ATP in there. This pink molecule here is going to represent our ATP. So ATP comes in and binds to the nucleotide binding domain 1. Okay, so ATP binds. Now, when it binds, what happens is that the nucleotide binding domain 2 and the nucleotide binding domain 1 then form what is known as a weak nucleotide binding domain 1, nucleotide binding domain 2 complex. Okay, so I will have to now redraw the picture to show this complex being formed. So I'll get another piece of paper. Okay, so what's going to happen now is that they're going to form a dimer, basically. They're going to form a MBD1, MBD2 heterodimer. So I'll show this in a picture. So you still have your transmembrane domain 1, and I'll try and speed up the drawing of the picture. And then you have your nucleotide binding domain 2. And basically this is going to have formed and the picture's probably going to be quite messy. So we've got the ATP molecule here, but basically it's going to have formed a heterodimer with the nucleotide binding domain 2, so I might as well draw that in now, and there's still this binding site free here. Okay, like that. So this is the nucleotide binding domain 1, and then nucleotide binding domain 1 obviously goes on to have the R domain here, and then R domain will go into, and this is where it's going to get messy, so I'm going to draw the other bit up here now. So I'll draw the next transmembrane domain down here. Okay, and then obviously this one then comes into the nucleotide binding domain 2. You then have the PDZ domain down here, and then finally the carboxyl terminus. So here we have the carboxyl terminus. So just to show that that's the same thing as what we had originally. What's now happened is that this has come and bound to this. It's formed a dimer, but the way it's all linked up as a polypeptide is still the same as in that picture. So let me just finish this picture off then. This is our ATP molecule here. We have our phosphorylation of the regulatory domain by protein kinase C and then also by protein kinase A. This is our nucleotide binding domain 2. Nucleotide binding domain 2. This is our transmembrane domain 1 and our transmembrane domain 2. This is our amino terminus over here. And anything else that we need to label, we won't bother labeling the PDZ domain. Right, we'll now label the entire thing that it's formed here. It has formed what is known as a MBD1, MBD2 heterodimer. And at the moment it's a weak one because it's only got one ATP molecule linking the two. We're going to put in a second one in a moment. Okay, so this is known as a weak MBD1, MBD2 heterodimer. Okay, so dimer just means that you've got two of them, and heterodimer means that they're different, basically. Okay, and hopefully MBD1, MBD2 should be pretty obvious. Right, so basically the formation of the binding together of this MBD1 and this MBD2 brings together the transmembrane domains. It brings those two, the transmembrane domain 1 and the transmembrane domain 2, closer together, but they're not close enough yet. What needs to happen is a second ATP molecule needs to bind there. So when a second ATP molecule binds there, then that turns this weak MBD1, MBD2 heterodimer. So once a second ATP molecule binds, it turns it into a strong MBD1, MBD2 heterodimer. So now it's a strong MBD1, MBD2 heterodimer. And once that has happened, and the two phosphorylations and the phosphorylation site of protein kinase A on this regulatory subunit is phosphorylated, then the channel will open and it will conduct anions such as chloride. So basically now what happens is it opens. Okay, so that's the set of events that leads to it opening. It is opened by ATP, so you need ATP to be quite high in the intracellular environment in order for ATP to bind in both of these sites and allow the formation of the strong MBD1, MBD2 heterodimer. And I want to emphasize again that if the ATP molecules bind and form this MBD1, MBD2 heterodimer, it will not cause the channel to open unless the regulatory subunit is phosphorylated by protein kinase A here. And in order to be phosphorylated by protein kinase A, you need to have been phosphorylated by protein kinase C. Okay, so it's a complicated mechanism of opening. Now, closing is no easier as well. So, basically, in the membrane, there are membrane-bound phosphatases. So I'll draw one of those here. So this is an enzyme, basically, up here. This is a membrane-bound phosphatase. So I'll just label it that. So this is a membrane-bound phosphatase. Membrane-bound phosphatase. And basically what this membrane-bound phosphatase is going to do is it's going to remove the phosphate group that protein kinase A put on the regulatory subunit. So it's going to take that off. So it's going to go down here and it's going to remove this phosphate group. So this is going to go, basically. And what you find is that if this happens, it does not cause the ion channel to close. The channel remains open. So channel remains open if this happens. Channel remains open. So removing the phosphate group from the regulatory subunit that was necessary, it had to be there in order for the channel to open, does not cause it to close, basically. So once you've formed this MBD1, MBD2 heterodimer, you don't need that phosphate group anymore to keep it open, basically. Once the channel is open and you've got your MBD1, MBD2 strong heterodimer, you don't need that phosphorylation there. So the membrane-bound phosphatase can remove that phosphate group quite happily. What causes the channel to close instead is not the removal of the phosphate group, but instead it is the hydrolysis of this second ATP molecule that is bound to the MBD2 molecule. Because basically, although I've drawn it as though the ATP is bound to both of them, in fact, it's a case that the ATP here, this first ATP, binds to the MBD1, changes the conformation of the MBD1, and then allows the MBD2 to bind. And the MBD2 then binds to an ATP molecule, this second ATP molecule. That changes its conformation and it allows it to form a stronger dimer, heterodimer, with the MBD1. So, to close the channel, what you do is you hydrolyze the second ATP molecule, i.e. the molecule that's bound to the nucleotide binding domain 2. So hydrolysis of second ATP. And as soon as that happens, as soon as the ATP hydrolyzes, that causes the channel to then close again. Okay, so that's pretty much all I've got to say for this video. We've done the structure of the Cystic Fibrosis Transmembrane Conductance Regulator, and we've done how it opens and how it closes.